Good evening. I'm Dick Meserve, the president of the Carnegie Institution, and I'd like to welcome you all to a Capital Science Evening. In November 1895, the German physicist William Röntgen noticed an odd glow on a fluorescence screen some distance from the vacuum tube he was using to study cathode rays. He didn't know what caused the glow. Curious, he redid the experiment and the glow reappeared. For the next several weeks, he holed himself up in his laboratory while he investigated what he was convinced was a new kind of ray. He called it an X-ray because he had no idea what it was, and of course, X denotes the unknown. His terminology remains with us. When Röntgen published his results a few weeks later, he included a photo he had made of the bones of his wife's hand. The world quickly took notice. Within a month, physicians, entranced by the medical possibilities of this new ray, used them to locate bullets in soldiers' bodies and to take pictures of broken bones and kidney stones. By the early 1900s, X-rays had become a part of cancer treatment. Well, the discovery of X-rays earned Röntgen the first Nobel Prize in Physics in 1901. Neither Röntgen nor his contemporaries understood, even after several years of study, exactly what X-rays were. They didn't know how or even if they were related to cathode rays or other kinds of rays. They didn't even understand whether they had the characteristics of waves or of particles. The answer that they had the properties of both would not come for many more years. Meanwhile, X-rays took a turn into the world of crystallography. The German physicist Max von Laue was convinced that X-rays were light waves with wavelengths much shorter than those of visible light. In 1912, thinking that the very short wavelengths of X-rays might be ideal to probe the spaces within crystals, von Laue discovered that crystals diffracted X-rays in regular patterns. His discovery, which earned him a Nobel Prize as well, led the English physicist William Bragg and his son Lawrence to develop a method to use the rays to calculate the exact positions of atoms within crystals. Just as they had for medicine, X-rays soon began to revolutionize the study of matter. X-rays revealed the size of atoms and crystals, the links and types of chemical bonds, and the atomic scale differences among the wide range of minerals and alloys. They also revealed the structure of many biological molecules, eventually including DNA. Today, X-rays remain a central means for studying crystal structure and behavior. The brightest or most energetic source of X-rays for atomic study is found at the SLAC National Accelerator Laboratory, a U.S. Department of Energy facility on the campus of and operated by Stanford University. The Linac Coherent Light Source at SLAC produces femtosecond X-ray pulses more than 1 billion times brighter than those from any other source. These ultrafast and ultra-bright pulses enable scientists to explore the atomic realm in ways that Röntgen and other X-ray pioneers could never even have imagined. Here tonight to tell us about the Linac Coherent Light Source is the former director of SLAC, Dr. Persis Drell. Dr. Drell served as director for 7 years, stepping down last October to resume her work as an astrophysicist and professor of physics at Stanford. Dr. Drell received her BA in mathematics and physics from Wellesley College and her PhD in atomic physics from UC Berkeley in 1983. She remained at Berkeley as a postdoctoral fellow while working at the Lawrence Berkeley Laboratory before joining Cornell University's physics department in 1988. In 2000, she became the head of the Cornell High Energy Group, and a year later she was named deputy director of Cornell's Laboratory of Nuclear Studies. 2 years later, she moved to Stanford, at which time she became the SLAC Director of Research, eventually becoming the director in 2007. Dr. Drell is a member of the National Academy of Sciences and a fellow of the American Academy of Arts and Sciences and the American Physical Society. Early in her career, she was an NSF Presidential Young Investigator. She has served on many advisory committees concerned with national research, facilities, and physics funding priorities. Dr. Drell will tell us tonight about the challenge of building the Linac Coherent Light Source and describe some of the first experimental results from this remarkable tool. Please join me in welcoming Dr. Persis S. Drell.
Well, thank you. That certainly ranks as one of the more complete and knowledgeable and very helpful introductions, actually. I'm going to use that, that introduction. Thank you. So I appreciate the invitation to come tonight, and I've really been looking forward to this talk. I'm going to tell you a story tonight about the X-ray laser that we have just built and turned on at SLAC, National Accelerator Lab, at Stanford University. And it is the story of a revolutionary new tool. And this tool is opening a frontier of science. And so I hope by the end of my talk, you'll have a sense both of how the laser works, why it is so revolutionary, and a glimpse at some of the early science that we are doing with it. But before I get started into the more technical details, I'd like to just make a general observation, and it's sort of a personal observation to set the stage. And it has to do with how we make progress in science. So I like to think of progress in science, the great leaps forward, as coming from two distinct directions. The one step I would call theoretical progress, and that's what happens when someone looks at the world from a different point of view or asks a simple question that no one has ever asked before. And maybe my favorite example of that is Copernicus in the 16th century when he asked the very simple question, what would the motions of the planets look like if we on Earth were orbiting the sun along with the other planets instead of having them orbit us? And that revolutionized our view of our solar system. Another example is when Einstein, as a teenager, asked, what would the world look like if I was riding on a light beam? And it led to his revolutionary theory of special relativity. But there's another category of great scientific progress and innovation that I would call experimental progress. That's when you create a new instrument that just revolutionizes your ability to look at the world around you. And maybe Galileo's telescope is one of the more familiar examples, when all of a sudden he could look at Jupiter and see the moons around it, or look at the pocked surface of the moon. And as I hope I will convince you tonight, the X-ray laser is such an experimental breakthrough that opens a new frontier of science, letting us look at the world in ways that we have never been able to see it before. Now, of course, sometimes you actually design a tool to look for new physical phenomena, and every now and again you design it for one purpose and it actually totally surprises you in some other area. And I don't know which of those two categories in the end will view the Linac Coherent Light Source, but I do believe it is revolutionizing and will revolutionize our view of the world in unexpected ways. So you've heard a little bit of the history of X-rays, which I think is a really excellent beginning. But to really make sure everybody's on the same page in understanding the power of X-rays, let's just start at the beginning with what X-rays are. X-rays, visible light, microwaves, radio waves, they're all the same thing. They're electromagnetic radiation. And the different names simply refer to the different wavelengths of the radiation. So visible light is the most familiar form of electromagnetic radiation. That's because your eyes are sensitive to it, so you see it all around you. But you're also pretty familiar with microwaves, right? You cook your supper with your microwaves in your microwave oven. They have a wavelength about 10 centimeters, whereas the wavelength, of the visible light is much shorter. Radio waves have wavelengths that are more like meters. And what's special about X-rays is their wavelengths are very, very short, about the size of an atom typically, of order, an angstrom is the unit we use, 10 to the minus 10 meters. Very, very, very small wavelengths. Now, X-rays have two very important properties that make them useful for science as tools. And I've just introduced you to the first one of those properties, which is the wavelength is very, very short. And Mr. Röntgen introduced us to the other property of X-rays, which was that they see through things, right? He could see the skeleton of his wife's hand. I'm sure you've all seen that picture. When you go to the doctor, you've fallen on the ice, your arm hurts, they use an X-ray to look inside your arm to see if the bone is broken. Here's my favorite X-ray. It's my son. He's about 8 years old, and he had swallowed a quarter, okay? And we rushed him to the emergency room, and they wanted to make sure that the quarter was safely in his stomach. So, nice straight spine, shoulder to pelvis, you can see his ribs, and yes, the quarter is safely in his stomach. It's actually a beautiful X-ray. The quarter's dead on to the X-ray beam. The focus is perfect. In the original, you can make out George Washington's silhouette. Okay. So X-rays let us see through matter. And because of their very short wavelength, on the order of the size of atoms and the spacing between atoms, X-rays can help us understand where are atoms in materials. So they tell us about the structure of matter. And they can help us understand where electrons are in materials, which helps us to understand what the electronic properties of materials are and to study them and ultimately to control them. Now, we've been using X-rays for decades, actually for a century, to study matter. And to this point, most of our studies with X-rays have been, let me use the word broadly, static. So we've looked at static properties of matter. We've looked at structure, where the atoms are. And it's deeply satisfying as a physicist that form is related to function. And when we understand where the atoms are, we can understand the functioning of materials. And just 3 examples that have had practical applications, X-rays were used to study the coatings on some night vision goggles that were being used in the Gulf. We needed to study what was going on because the coating on the night vision goggles was deteriorating in the very dry atmosphere of the desert. They used X-rays to probe the structure of the coating, diagnose the problem, improve the coating, improve the night vision goggles, which are now back in service. Another example is that X-rays were used to determine the structure of an mutant enzyme that is activated in melanoma. But from the structure of the mutant enzyme, a treatment was devised that could block the progress of the melanoma, unfortunately only temporarily. But for stage 4 melanoma patients, it bought them time measured in months, not a cure, but progress. And then a third example was that by determining the structure of the H1N1 flu virus, one was able to design some antiviral treatments to minimize the impact of the flu. But these were all studies of structure, static studies, static structure. We've been doing this for decades. And the new frontier, the new dimension is time. What I'll be talking about is not only being able to look at atoms, where they are, but in fact what they are doing. On the time scales, they're doing interesting things. That is the new frontier, atomic scale dynamics. Now, I'm going to use an analogy to explain both how important dynamical information is and to illustrate a little bit about how we're going to use it. And the analogy has to do with galloping horses. So, if you were to go and look at paintings in an art gallery or on Wikipedia, that great reference, of galloping horses, you will note that before about 1878, people had very imaginative ideas of how galloping horses actually did their galloping. And the reason is that if you look at a galloping horse, they're moving so fast, their legs are moving so fast, you can't actually tell what's going on. And so painters use their artistic license to imagine whether all 4 feet of the horse left the ground or not, and where the feet were when they left the ground. And it was just not known until 1878. There was actually a bet put forward by Senator Stanford, who founded Stanford University. He had a lot of horses. He was a betting man. And the bet was to answer the question, do all 4 feet of the horse leave the ground when it gallops? And a very clever guy named Edward Murbridge designed a camera with what was then the fastest shutter speed known, 0.001 seconds. He set up a series of tripwires, took a series of pictures of the galloping horse, and if you put those freeze frame stop action pictures together, you can immediately see the answer. Yes, all 4 feet of the galloping horse leave the ground, and they leave the ground when the 4 feet are under the body of the horse. So the fast shutter speed, or equivalently, it could have been a fast strobe, was able to give sufficient dynamical information to answer that question. So now I'd ask you to imagine what you could do if you had an X-ray camera equivalent with a shutter speed equivalent of 0.000000000000010 seconds. Okay? First, that's a really short time, okay? It's 10 femtoseconds, and to give you a sense of scale, light travels pretty fast. It takes about 1.5 seconds for light to get from the Earth to the moon. Okay? It's going pretty fast. In 100 femtoseconds, light travels about the distance that's the same as the thickness of a human hair. So 10 femtoseconds is really fast. Well, if you had such an X-ray camera equivalent with that kind of shutter speed, you would be able to take stop-action pictures of atoms on the time scale that they're doing interesting things. You would be able to watch chemistry atom by atom, step by step. That's the frontier that we opened in 2009 when we turned on the X-ray laser at Stanford. So, what's an X-ray laser? And in fact, this isn't just an X-ray laser, it's an X-ray free electron laser. Okay. So the first thing I have to say about the X-ray free electron laser is it's not your normal laser. So this laser pointer, as you can see, is a normal laser. Okay. It's light amplification by stimulated emission of radiation. And most of the lasers you've dealt with, and you deal with lasers all the time, right? Every time you go to the checkout at the grocery store, you've got a laser scanning your barcode. Those are all light amplification by stimulated emission of radiation. The free electron laser uses different physics, if you will, to generate the radiation. But the output is very similar to the radiation from this laser pointer. It's very bright, it's coherent, it's collimated, and so it's, it walks like a duck, it quacks like a duck. We're going to call it a duck, even though it's not laser in the traditional sense of the word. The output of the free electron laser is laser-like. So the free electron laser is not a new concept. The first free electron laser was built in 1977. The father of the free electron laser is John Mady, a very interesting character, I have to say. And the first free electron laser that he built was in the infrared. It was 3.4 micron wavelength. And since 1977, there have been literally dozens of free electron lasers built around the world, mostly in the infrared and in the visible. In 2005, the first free electron laser was turned on in the far ultraviolet, but no one had gone into the X-rays. It was viewed as quite challenging. The free electron lasers that were built in the decades since they were invented by John Mady were non-trivial to operate. They had a reputation for being somewhat finicky. But scientists like a challenge. And in 1992, a very smart accelerator physicist named Claudio Pellegrini recognized that if you had a powerful electron accelerator, and I'll explain why you need a very powerful electron accelerator, but if you had a powerful electron accelerator, you could actually make an X-ray free electron laser. And that's where that concept was born, and ultimately the project was funded. It was a big deal. One's talking about a very large facility, $400 million to build. This is not a little tabletop device, I want to tell you. But the recognition from the beginning was that if you could make this work, if you could get an X-ray laser with very, very short pulses, you would see the world in ways no one had ever had access to before. So the concept behind a free electron laser is illustrated here. You need a bunch of electrons. In the end, actually, it's all about the electrons. You need to start with a very, very intense bunch of electrons that are in a really, really small bunch. So talking about 1 billion electrons in sort of micron-sized dimension bunches. You take that bunch of electrons and you put it down a very long undulator magnet. An undulator magnet is a magnet where the poles flip about, in our case, every few centimeters. So it's north, south, north, south, north, south. And by very long undulator magnet, I mean like 100 meters of undulator magnet. And what happens is the electron bunch wiggles back and forth. It's a relativistic bunch, wiggles back and forth under the influence of this varying magnetic field. And it starts to emit radiation. The fields of the electrons and the fields of the emitted radiation are so intense that they self-interact. And the radiation starts to affect the electrons, which starts to affect the radiation it emits. And you have a phenomenon called microbunching. They order the electrons into little microbunches within the big bunch, and then the ordered electrons start to radiate coherently. So I'm actually going to show you a simulation of how this works. All that goes into the simulations is a lot of electrons and electricity and magnetism. And here it is. You start with a bunch of electrons going through an undulator magnet being oscillated back and forth, and then as you watch, the microbunching starts to happen as you get this self-interaction between the radiation that's emitted by the electron bunch and the electron bunch itself. And you get the microbunching, and then the microbunches radiate as if all of this charge was in one single electron. So that's sort of the concept of how the free electron laser works. As I say, it's all about the electrons. You need superb quality of the electron bunch. It needs to be accelerated to, in our case, between 5 and 15 GeV. That means if you use E equals mc squared, these are relativistic particles, that the electrons have an energy that's about equivalent to the mass of between 5 and 15 protons. So it's a fair amount of energy, but not world-breaking or anything. The energy of the electrons will determine the energy of the X-rays that are emitted. And in our case, it requires that you have to pack 6 billion electrons into a 30-micron sphere. And that's hard, okay? That's really hard. This is actually what the electron bunch looks like at the end of our linear accelerator. And here's a picture of the linear accelerator that, in fact, the whole lab, which is where this all happens. So for those of you who have visited Stanford University before, Hoover Tower and the campus with all the red tile roofs is about 2 miles down the road here. The Pacific Ocean is on the other side of those hills. Sand Hill Road, venture capital center of the world, is right on the border of our site. And this is the laboratory, SLAC National Accelerator Lab, sitting on 422 acres of Stanford land, leased to the government at no fee, one of the great bargains still out there. The 2-mile-long electron accelerator was built actually starting in 1962. It's a middle-aged lab. I was going to say old, but these days I call that middle-aged. We just had our 50th birthday. Originally, this 2-mile-long linear accelerator took electrons, accelerated them to the highest energies available at the time, smashed them into protons and neutrons, and that's where the quarks were discovered. Okay, that was a different era and a different mission. The frontiers of particle physics move on. The center of the universe for particle physics right now, as you all know, I think is at the LHC and the discovery of the Higgs. But this beautiful 2-mile-long Linac works still like a charm. Probably the electron bunches out of that linear accelerator are better understood than any other electron bunches in the world. And Pellegrini's genius was to say, I could take a third of that 2-mile-long Linac, just the last kilometer. I only need the last kilometer. Put an injector in, accelerate the electrons to between 5 and 15 GeV, then put them through an Undulator Hall that's actually under this hill for reasons I'll explain in a moment. That's where the Xrays will be made. And then the experiments using those Xrays will be carried out at the two interaction halls, the near hall and the far hall. So here's a little simulation of how it all works. These buildings up on the surface house the power to run the linear accelerator. There's a conventional laser up on the surface that sends laser pulses down to create the electron bunches by the laser hits a specially prepared copper cathode, kicks out a bunch of 1 billion or so electrons, and that bunch of electrons, they are kicked out at about 120 times a second. It gets pre-accelerated, and then at the 1 kilometer from the end point, put into the same accelerating structures that were originally built in 1962 for the linear accelerator. We play some tricks to shorten the electron bunch and make it small, the tens of micron size that we need to, and the amount of energy we give the electrons depends on what energy X-rays the experimenters at the other end are going to want. But we have quite a wide range of energy of X-rays we can deliver the experimenters, determined by how much acceleration we give them in the Linac. We then allow the electrons to drift for a while, making sure they stay focused, and then they go into the Undulator Hall, and this is where the magic happens. This is 100 meters of precision undulator magnets. The electron bunches are being wiggled back and forth in those undulator magnets. They're in a temperature-controlled hall under the hill. The hill is very nice for keeping the temperature very, very constant. The floor is stabilized. And as the electrons wiggle back and forth in the undulators, they start to generate radiation. The radiation starts to affect the electrons. The microbunching occurs, and you start to have production of X-rays. At this point, you have a pulse of X-rays that's traveling with the electron bunch. Again, these pulses are coming at 120 times a second. And once we've gotten out of the undulators, the electrons have done their work. We don't need them anymore. And so we have a powerful magnet that kicks the electron bunch down into a dump in the ground, and the X-rays stream free now into the experimental areas. The experimental areas, there are 6 hutches, 3 in the near hall and 3 in the far hall, and typically we deliver to a single hutch. We're just learning how to deliver to 2 hutches simultaneously, and we will have another experiment ready to take beam, so we'll have a 12-hour on for one experiment, 12 hours to the next. Okay. Now, this is what the Undulator Hall actually looks like, and when we first sent electrons through the undulators, we did not know if this was going to work. Okay? There were quite a few people in the community, in fact, who predicted we would get a lot of intense X-ray radiation, but we wouldn't get lasing because the tolerances were too high and for whatever other reason. So we were a little cautious, and we got everything ready, and in April of 2009, we started the lasing campaign. I went to the control room that afternoon, and the atmosphere was, I mean, you could have cut it with a knife. Everyone was very tense, you know, they were ready, but we thought it was going to be a long slog, and this is what it looked like in the control room. This is Paul Emma. He headed the commissioning. He led the commissioning team, and they're looking slightly worried. This is John Galida, the project director, but this was the moment of truth. That's the first sign that we actually were seeing lasing, and this is about 10 o'clock. Right? Now, I had gone home and gone to bed. Lab directors go to bed early on Friday nights, and I thought they had weeks to get this thing to work. I got the phone call. I was woken up. We have a laser. It was a miracle. There's just no other way to say it. It worked. It worked from day one. And the performance of the X-ray laser exceeded expectation in every possible dimension. It's hard to describe how much fun that is. If you like numbers, these are the numbers in terms of how we expected it to perform, how it's currently performing. We can make the laser. It's very flexible. It's turned out to be much more flexible than we thought. We can do all sorts of things with it. But the bottom line is this is what X-ray sources could deliver in terms of brightness before the LCLS turned on. It turned on 1 billion times brighter, 9 orders of magnitude. That's what opens a new frontier. So I'd now like to tell you a little bit about some of the first science. And there's a lot of science going on now. I would say we're still in a phase where we are learning how to use this facility. It is, for the first 3 years, it was absolutely unique from turn on. Now there is another X-ray laser that has turned on in Japan. Doesn't have quite as much energy, but starting to do very interesting experiments. We actually really welcome that because at the moment, you can only deliver beam to a finite number of people, and the progress of science is actually slowed by the fact that we can't get enough people on the beam lines. Another X-ray laser will turn on in Germany. It's scheduled to start in 2016. I think they might be about a year late. And there are at least 2 more under construction now with more to come. It's turned out, now that people see that the technique works, to be a really attractive target for a lot of scientific communities who want their own. Just a word about the way we do science at this facility. We operate this facility for the world. So we build the experiments, we build the laser. We have scientists at SLAC and at Stanford who are very eager to use the facility, but they have no preference over anyone else in the rest of the world. There's a proposal process. People submit proposals. There's an external review committee that comes in. The best science wins, independent of where it comes from. And if you are a winning proposal on the LCLS, you get beam time. We help you with your experiment all free of charge. Okay? It's amazing, but it's the way to deliver the best science. Right now, we turn down 3 to 4 proposals for every one we accept because the demand is just really tremendous. So I'm going to talk about 2 specific experiments. As I say, it's been selected from a range of the first experiments. There was actually just another press release today of a very exciting experiment. If I have time at the end, I'll talk about it, but let me focus on these 2. So the first thing we needed to do with the LCLS was to find out if the concept of taking stop-action pictures, if you will, at the atomic timescale was going to work. Because there's this slight problem when you take something and you put it in the laser beam, which is if you drop a little object, a little nanocrystal or something in, and the X-ray laser comes along, you scatter the X-rays, you get the diffraction pattern, but you just blew up your sample. Okay? So you don't get to take a second picture. You got to get a second sample. So it's really the equivalent at the atomic scale of, can you take the picture of the light bulb you just dropped before the light bulb breaks? So we call that technique probe before destroy, and I want to explain to you how that works, and it does work, and it's actually turned out to generate a whole scientific branch of investigation on its own. But then what we're really interested in doing is once you show probe before destroy works, can I now use this technique to study chemical dynamics? Can I take a multi-step chemical process, use probe before destroy, take a series of stop-action pictures, put them together, and make a movie of chemistry in action? That's the goal. So we start with probe before destroy, and interestingly, almost a decade before the X-ray laser ever worked, there was this seminal paper published in Nature. It was a thought experiment. And it said, if you wanted to do an experiment of, in this case, maybe a single particle or a single molecule at an X-ray laser, here's how you'd do it. You'd have the X-ray pulses coming in. You'd drop your particles one by one. The X-ray pulse would hit a particle. It would destroy it, but you could capture the image before it was destroyed. That was the concept. In sort of real time, this is what it might look like. One pulse, one laser pulse, one particle. You capture an image, and then because each image is sort of noisy and doesn't have a whole lot of information, you're going to have to combine a whole lot of those images in order to reconstruct what the original particle looked like. Now, analogies are never perfect, but this reconstruction is non-trivial, and the best analogy I can give you is it's like taking a large misshapen rock and dropping it in your swimming pool, and then you look at the ripples that hit the edge of the swimming pool, and by doing that a bunch of times and very carefully measuring those ripples on the edge of the swimming pool, you're supposed to be able to reconstruct the shape of the rock you dropped, okay? But people can do it. This is what the actual experiment looks like. We use, initially, we don't use single particles. We use nanocrystals, typically maybe a few hundred or a few thousand molecules arranged in a tiny crystal. A crystal that's too small that you could use it to do structure determination at any one of the conventional sources. Maybe it's a material that doesn't grow big crystals, which is why it's a good target for the X-ray laser. But you drop the nanocrystals, they intersect the X-ray beam that's focused on them. You have a CCD detector like what's in your camera, well, not exactly like what's in your camera, but conceptually like what's in your camera, and you record the diffraction patterns. This is what one of the single-shot diffraction patterns looks like. We started, of course, with structures that were known to make sure that we could actually reproduce atomic structure level information that had already been generated at other X-ray sources. But then we started to move on to trying to see if we could determine structure of substances, crystals, that one couldn't image by traditional means. And this is one such example. It's the first published result on a structure that hadn't been previously solved. That was a single shot image. This is what you get by combining many, many shots. And it's the enzyme that is key for the survival of the parasite responsible for African sleeping sickness. This is the structure that was determined at the LCLS. To be perfectly honest, they didn't do the full de novo structure. There was some information about the structure before, but they didn't have a complete picture of it, and LCLS was able to fill in, using existing information, some really key elements of that structure. And this is something that, again, form is related to function. Once you understand that structure, you now have a shot at designing something that can block that enzyme and maybe make contributions to what is really a terrible, terrible disease. So that was probe before destroy, and it works. I mean, there was a lot of uncertainty. Would you damage the object in the process of measuring it? So they did quite a few studies to, in fact, show that that was not the case. Here, the fact that those pulses are so short is what is absolutely key. And there's now a whole industry, if you will, that is now focused on taking substances that cannot be imaged by conventional means, by conventional X-ray sources, and seeing if we can determine their structure now using this new technique. But we're still talking about static structure. So now we want to take the next step. Can we take this probe before destroy technique, image a multi-step chemical process, and make a movie of it? So I was talking about this on the Hill once, and one of the staffers says, "Oh, you're going to make Photosynthesis the movie." And I thought, yes, that's what we're going to do. So you all learned about photosynthesis in what, 5th grade? Plants, it's the engine of life. Plants take carbon dioxide from the atmosphere, water, presumably from the ground. They convert light energy into then sugars, and they release oxygen. That's the engine, okay? It's a non-trivial process, okay? And this membrane protein, photosystem II, is key. It's the one that splits the water, which is hard to do. So that's what we want to study. And what happens in the photosystem II is it's called the Koch cycle. It's a 4-step process where the light comes in, it absorbs a photon, it moves to a new configuration, it absorbs another photon from the sun, moves to yet another configuration, absorbs another photon, moves to another configuration, absorbs another photon, and the water is split. Okay? So the idea here is image, excite, image, excite, image, excite, image, excite, image, and see what you get. Okay? Because it's the configurational changes between those states that we're after. Okay? We haven't gotten all the way there. There are 2 teams of scientists furiously pursuing this problem. They're not talking to each other. It's okay. A paper was just published 2 weeks ago, which showed the structure. They got the structure of S2 and S1, along with actually some other very interesting spectroscopic information. They didn't get S3, and they didn't really have the resolution on S2 and S1 that one would want. But what they were able to show is that there are no major configurational changes at this point, they were not expected. This is the step where the significant configurational changes are expected to happen. We don't have that yet. The second team is writing a paper. I know they're writing a paper, but I don't know what's in it. So I can't share that with you. This is actually what the data looked like from the first team. This is a single-shot image of photosystem II nanocrystal. This is what the little nanocrystals look like. They're green, this plants. And this is what the electron density maps look like for that photosystem II. So very, very exciting. What have we learned at this point? Well, we know probe before destroy works. We've demonstrated that. We've all viewed that as the first killer application of the X-ray laser, because now we can take substances that can't be made into big crystals and have their structure determined by conventional means, and we have a shot, at least, of making nanocrystals out of some of them and determining their structure using the X-ray laser. We've shown we can do it, and the biologists actually are starting to line up with their samples to see if we can make progress here. Just to put some reality, though, in the situation, these experiments are hard, and one of the technical challenges we're facing right now is not that complex, huge X-ray laser. It's the tiny little nozzle that drops the nanocrystals into the beam is one of our big challenges right now, because every biological sample is special. It wants its own nozzle and its own delivery mechanism, and we're struggling with being able to automate that in a way that can really get those samples through. That's where one of the bottlenecks is now. But no one questions this is going to be a major, major impact from the X-ray laser. Chemical dynamics after excitation is being studied. That's the photosystem 2 example from photosynthesis. I believe free electron lasers will solve photosynthesis. I think it's very, very exciting. The press release today was also, I would say, in this category. It was a model system, a toy system, looking at a catalytic process on a surface, a ruthenium surface, a carbon monoxide molecule, put on the surface, and then watched as it desorbed from the surface, discovered that there was an intermediate state people hadn't known about, that the carbon monoxide sort of hung around in for a while. Lots, I think, to look forward to in that area as well. Now, next steps, can we image a single particle, not a nanocrystal? Because there's probably stuff that doesn't even make nanocrystals that we want to know the structure of. Could we actually image a single molecule, or maybe a single cell or a single virus? Well, people are already trying that. This is a single-shot image of a Mimi virus, a fairly large virus. The real challenge for these samples is with the nanocrystals, we use the crystalline structure to fix the molecules in a rigid structure, all in the same orientation. But when you're dropping them one by one, you have a heterogeneity of your sample, that each cell or each molecule is going to be in a different orientation in the beam, or maybe the cell is going to have its innards rearranged. And there's a lot of mathematics and theoretical study going into can we solve those problems? I think the answer is going to be yes with enough data, and will we have enough data, and will our detectors be noise-free enough to really make it happen? But again, very, very exciting, very active area, because people see that as a real target for new science. So I'm going to end here. I think the near term is just incredibly exciting. We are having so much fun with this new tool. The LCLS has been so successful that we are actually expanding it now to double its capacity. After all, we only used 1 kilometer of that 2-mile-long Linac. There's 2 kilometers left. So we're taking the middle kilometer, thanks to the generosity of the DOE, and turning that into, we'll build another undulators, another pair of undulators and more experimental stations. So more science will be done using what we've learned from the first incarnation of LCLS to even improve it. New facilities are turning on. As I mentioned, the X-ray laser in Japan is operating. Germany will be operating, I think, on the 2017 timescale, and then Switzerland and Korea by the end of the decade, and more to come. A new scientific frontier has been opened. There's just no question. It's a time of extraordinary opportunity as we go from structure to really dynamics at the atomic scale. And I think what we all believe, all of us who are involved in the science of this project, is that the biggest surprises are really still out there, and we don't know what they are, and that's what's going to be what's really fun in the decade to come. Thank you very much.
Well, Persis has obviously provided us with a tour of a fantastic new tool for science. We do have an opportunity for some questions. There's a microphone here, and I'll invite people to come to the microphone. Please.
Thank you. I've worked on several projects over the years that seem to fit right in with this. One is the energy transduction from cytochrome C. Another one is energy transduction using bacteria rhodopsin. And finally, one that is not an energy transduction thing, but related, the conformation of hemoglobin as it is oxygenated. That is, the human hemoglobin can take 1, 2, 3, or 4 oxygens, or none.
Yep.
And it would be very nice to know at what stage the big conformations occur. And I don't think anybody's really sure of that yet. So do you have any proposals in the pipeline about these things?
Well, so I think that what's going to be key is if we can get the single particle imaging, single molecule imaging, if that can be made to work, then I think we have a shot at those issues. The other thing, the other strategy is going to be, can you somehow, I don't think hemoglobin crystallizes, but can you use some clever trick to put it in some kind of a crystalline structure, because then one could start to attack it even sooner.
The answer is yes,
you can crystallize. Then you have if you can crystallize it, then it's a matter of you may not be so far before being able to attack some of those problems. Please do.
Please.
This might be a little more of an elementary question, but you mentioned that there was some uncertainty once the facility was constructed whether or not you could get the X-rays to laze.
Right.
I didn't quite understand, after the X-rays are generated by the micro bunches of the electrons,
right,
what is involved in focusing those into a laser-like beam?
Okay. So the answer is they come out, they're generated by this very tiny electron bunch, and they come out coherent and pretty, going straight, collimated. We do then focus them in the experiments, but the coherent light is generated by the electron bunch. There's no mirrors involved. Okay, it's called self-amplified spontaneous emission because, in fact, it's like a single pass laser. It's a little unusual. You can focus X-rays, and we do focus them on our samples. It's not so easy. You have to either use beryllium lenses or other tricks, but you can focus them. But that's not necessary for the lasing. The electron bunch does all the work for us, which is a good thing.
Let me ask a question
Sure.
that has to do with experimental technique. As even in your nanocrystal example, you're dropping these pellets down, and I presume you have to have the orientation of the crystal be constant from pellet to pellet,
Okay.
so that you have a diffraction pattern that you can interpret. How do you do that?
That's a great question, and the answer is no, you don't. We get so much information on a single shot, we can index the pattern shot by shot.
Oh.
That was one of the early miracles, okay, that we could do that. So, but that highlights the challenge of the single particle imaging where we don't have that ability to index the pattern because there's no crystal there. And so then the heterogeneity gets you. Now, there's a tremendous amount of work going on in a variety of labs to think of ways of aligning samples, because if you could even just remove a few degrees of freedom from a particle, you could probably get it. But in the case of the nanocrystals, we get so much information on each shot that we index them shot by shot.
Please.
You mentioned during your presentation it takes about 12 hours to conduct an experiment.
Oh.
It's just that data acquisition or starting from generation of the laser?
Okay, so
Elaborate.
typically what we do is we schedule an experiment. Somebody writes a proposal. We schedule their experiment. We schedule them maybe 3, 4, 5 shifts. But people can't do that 24 hours a day. So after some experimentation, we settle on a schedule where people will get 12 hours of beam time, and then they get 12 hours down. Somebody else takes the beam, 12 hours of beam time off, and they get that for 5 days. And that's a typical, what a typical proposal will get. Some proposals, more complex setups, can get more time. Simpler ones can get less time. But the 12-hour cycle just came from sort of optimizing what the human body can take, and also with if we're alternating between 2 experiments, there's a certain overhead. One experiment wants 1.5 angstrom X-rays. The next experiment wants 800, maybe 800 eV or something. So you have to change it. Somebody wants long pulses, somebody wants short pulses. So there's an overhead of changing the accelerator that we try to minimize. And it works out pretty well. The experimentalists actually like having 12 hours to take data and then 12 hours. They typically have 2 teams to look at the data, figure out what's going on with the experiment, maybe make some tweaks, and then come back. Very often, at the end of their run, instead of doing 12 on, 12 off, 12 on, 12 off for the 5 days, they'll do that for 3 days, and then they'll arrange with their, the other team that their last run is a 24-hour run where they just go for broke.
Please.
Some years ago, I heard about the possibility of lasers being used to fuse. Is there any possibility that this technique might be applied for controlled fusion?
Okay, simple answer is no. We're nowhere near the energy densities that's needed. The National Ignition Facility at Livermore National Lab, they are attempting laser-initiated fusion. So it's an ongoing area of research, but we're nowhere near those densities.
Well, Persis has really provided us with an idea of a fascinating new tool that is going to be an enormously powerful vehicle for doing a lot of science. Please join me in thanking her for a great lecture. Thank you all for joining us. We're now adjourned.